Hey everyone, today we will talk about molecular biology, which is one of the topics of biochemistry in USMLE. And first I want to start by showing you a big picture of it because our human body is an organism which contains of organ system, and every organ system consists of particular organ, and every organ consists of tissue, and every tissue consists of particular cells, and every cell contains particular organelles. And at that level we will talk about molecular biology. And first we will talk about what is our genome, because our genome is all of the DNA in our body together. And when we say DNA, it usually exists in certain form depending if we need it right now or not, if we need it like to be actively expressed or not. So when it's compact, like when it's densely compact, it is inactive, and it has to be compact in certain points. And when we need a certain gene to be expressed, we just loosen this compact structure to make it more loose and be able to express later. So here we have our genome, which consists of 23 pairs of chromosomes in each somatic cells, because when we talk about genome of particular organism, we talk about somatic cells. And when we talk about sex cells, it is not defined particular organism, because it makes its function to make other organism, new organism, to multiply. So here we have our euchromatin when it's loose, and heterochromatin when it's compact and inactive. And actually heterochromatin consists of heavy methylated DNA and deacetylated histones, which is lowers our transcription activity. So it is what is DNA function, not only transcription, but it have to divide in order to grow and repair itself and reproduce as well. So when we say about grow and repair, we refer to a replication process, right? When two identical cells are formed. And when we say reproduction, we use replication in the interphase, and also produce other cells, but with a haploid number of chromosomes. So also, second function is to maintain biological processes. In biological processes, we need some biological molecules and organelles, and all of these molecules and organelles consist of proteins. So first function, which in order to divide, in order to make more cells, we have to undergo replication first. Make two identical DNA first. And in order to make proteins, we have to make first RNA, which is matrix for synthesizing proteins. It also has different forms of RNA, like mRNA, it is matrix, rRNA, it is in part of ribosome, which also will take place in order to synthesize protein, and it is tRNA, which also will bring some amino acids to create this polypeptide chain, and after proliferation it will become a protein. So in order to grow, repair, and reproduce, we have to undergo cell cycle in order to make two cells. And every cycle, like every cell cycle, consists of interphase, which includes G1, S, G2, and metaphase, which consists of several steps. We will discuss in this several steps in our next lecture, but today we will be discussing about interphase, and after mitosis or meiosis, we will go cytokinesis, where two separated cells will be formed. So when we talk about interphase, what we're referring to, we're referring to preparation for a cell division, where a cell will grow and genome will duplicate, and then we will undergo its division. So in G1 phase, G1 and G2, the letter G refers to gap, where cell is not divided. We have also G0 phase, where cell also not divided. But first we will talk about G1, which is where matrix for DNA replication will be undergo check out for duplication for some mistakes, and after that it will undergo replication, where we will have two chromatids of one chromosome, because when we not are undergoing any division, our cells usually have like stick-like form. But when it will undergo S phase and interphase, it will contain two chromatids of one chromosome, and it's still one chromosome, but it undergoes replication, and it now has two DNA structures, but it's still one chromosome. And after that, we have to check our newly synthesized DNA to any mistakes, duplications, and if we have any, we have to repair these mistakes. And after that, we will divide these genes, like we will make two separated chromosomes. Here we have one chromosome, here we have two separated chromosomes after it undergoes mitosis, but still we have one cell. And after cytokinesis it occurs, we have two separated cells. If it's mitosis, we have identical cells, like diploid cells. If it's meiosis, we have different from maternal cells, which contains haploid numbers of chromosome. And now we will talk about G0 phase, which not all cells will be entering, because some cell will go cycle after the cycle, and this cell will be called labile cells, for example epidermis, and some cells will go to G0 phase, but they won't be there forever. Like when we have some injury or surgery, for example, you know about liver transplantation, where part of the liver will be transplanted to the recipient, and it will grow and it will function like normal liver. So depending whether we have injury or not, it will divide and produce its cells to maintain normal function. And when we have like cells which will go to G0 phase forever. It will never produce same cells after injury. It will like repair, of course, but it will form fibrous tissue. It will form scar after it will be injured. For example, cardiac muscle, after we undergo infarction, we have scar tissue formed. If nerves are damaged, also we have scar formed in this place. It will never function as normal neuron cells, or it will never function as normal cardiac myocytes. And our skeletal muscle as well will go to G0 phase forever. Here we have our table with labile tissue, which is for example skin, bone marrow, and it is like continuously regenerating, and stable tissue which goes to G0, but it can be undergoing a cell cycle. Depends on whether it has injury or not. It is liver, kidney, and pancreas. And when we talk about permanent tissue, as I said, it is terminally differentiated, non-proliferative, and it is neurons, cardiac muscles, and skeletal muscles. So what cells undergo when it's in G0 phase? G0 means it is in gap phase, where it is not dividing, but it is normal. It performs all of its functions. For example, if it's cardiac cell, it will contract, it will conduct all of the impulses. If it's neuron cell, it will be in G0 phase, but it will conduct and make impulses, right? Etc. Depending on if it's somatic cell or if it's sex cell, we have particular number of chromosomes. So if it's somatic cell, it will contain diploid number of chromosome. I mean 23 pairs or 46 chromosome. When we talk about gametes or about sex cells, it will contain diploid number of chromosome. I mean 23 chromosomes, and it will be produced by undergoing two steps of division, like meiosis 1 and meiosis 2, and it will form diploid number containing cells. And after mitosis, when we talk about somatic cells, it will contain identical number of chromosomes as its mother did. Like here we can see like maternal cell and its daughter cell, which contains same amount of chromosome. I mean 2 pairs of chromosome. And here you can see X-shaped chromosome, because it how it looks after interphase occurred, right? And here you have same amount of chromosome, but it is like rod shape, because it is already undergone division. So here you have also same process as in mitosis, and after that you have undergo same process for the second time, and now your cell look like this, like diploid number, different from maternal, because maternal contained 2 pairs of chromosome. Now you have 1 pair of chromosome. So here you can see spermatogonium or gonium contains 4 chromosomes. It is not yet gamete. It is not yet mature spermatocyte or ovum, which contains haploid number of chromosome. It is oogonium and spermatogonium, right? So after it undergoes all of the processes of meiosis 1 and 2, it will contain haploid number of chromosome.